IB Bio Molecular Biology Part 5 has its focus on lipids, structure, and function. The essential idea is compounds of carbon, hydrogen, and oxygen are used to supply and store energy. Here is the outline of all the movies available for studying IB molecular biology. Use this slide to reference the part of the topic that you need to review. This movie is focused here. Let's jump in with an IB syllabus statement that you've seen before, but modified to reflect lipids. Outline the role of condensation and hydrolysis in the relationship between 3 fatty acids and glycerol, the formation of triglycerides. Now, even with some new language embedded into this statement, you should immediately feel comfortable with it. Condensation synthesis, which is an anabolic energy storing reaction, will be involved when 3 fatty acids join with glycerol to form a triglyceride. Hydrolysis, a catabolic reaction, releasing energy in the process, is the reaction by which triglycerides are broken into glycerol and 3 fatty acids. Here are the first important IB syllabus statements of this movie. Draw the structure of a fatty acid. Outline the role of condensation and hydrolysis in the relationships between fatty acids and glycerol and triglycerides. Here is a fatty acid. A fatty acid is a long chain of CH2, CH2, CH2 all the way to the end where there's a CH3. The carboxyl end is joined to glycerol in condensation synthesis. A triglyceride is formed when 3 fatty acids are joined to 1 glycerol through condensation synthesis. Water is removed here and here and here, 3 total, when these fatty acids join glycerol. In this diagram, you can see the condensation synthesis as 1 fatty acid is joined to the glycerol with the removal of water. Enzymes catalyze all condensation reactions and all hydrolysis reactions. 1 fatty acid will join glycerol here, a second fatty acid will join glycerol here, and a third fatty acid will join glycerol here. The carboxyl end of the fatty acid joins with the glycerol with the removal of water, condensation synthesis. In this diagram, fatty acids are represented in a shorthand form, the carboxyl group. The number of CH2s is variable, represented as N, and a methyl group at the end of the long fatty acid chain. You can see 3 fatty acids joining with glycerol in an enzyme-catalyzed condensation synthesis reaction where 3 water molecules are removed. Notice that the carboxyl end of the fatty acid is joined to the glycerol with the removal of water. At the top of this diagram is the addition of a fatty acid chain to glycerol with the removal of water. Take note of which end of the fatty acid joins to the glycerol with the removal of water. In the lower portion of this image, we have a triglyceride. 3 fatty acid chains anchored to glycerol. Triglycerides are nonpolar and have a lot of energy per gram relative to other biomolecules. Here is a triglyceride, 3 fatty acids anchored to glycerol. Triglycerides have various roles in energy storage, insulation in mammals, and the waxy cuticle on insects and on the leaves of plants. Glycerol and 3 fatty acid chains joined through condensation synthesis to form the lipid or the triglyceride. Triglycerides or lipids are broken down by hydrolysis into 3 fatty acids and glycerol. The next IB syllabus statement is distinguish saturated, monounsaturated, and polyunsaturated fatty acids. Unsaturated fatty acids contain a double bond, at least one, along the hydrocarbon chain. As a result of the double bond between these 2 carbon atoms, there are fewer hydrogen atoms that comprise the hydrocarbon, thus the term unsaturated. Saturated fatty acids, as seen here and here, have no double bonds. Saturated fatty acids have the maximum number of hydrogen atoms possible, thus the term saturated. Unsaturated fatty acids could have a bend in the hydrocarbon chain as a result of the double bond. This fatty acid has one double bond, so it's called monounsaturated. In this diagram, there is a saturated fatty acid with no double bonds, a monounsaturated fatty acid with one double bond, and a polyunsaturated fatty acid with more than one double bond. Animal fats are often comprised of saturated fatty acids, while plant fats contain mostly unsaturated fatty acids. Fish also have many unsaturated fatty acids. Here's the next IB syllabus statement. State that unsaturated fatty acids can be cis or trans isomers. When fatty acids have a double bond, the structure of the molecule can take one of two forms, the cis form or the trans form. In the cis configuration, the double bond produces a bend in the hydrocarbon chain. In the trans configuration, the double bond does not produce a bend. This is a graph that shows the relationship between cardiovascular disease and the implementation of a trans fat ban in commercial foods in New York State counties, 2014. We have years to implementation of the trans fat ban here on the x-axis and cardiovascular disease-related deaths per 100,000 here on the y-axis. The trans fat ban was implemented in some counties in New York State, and that is shown here in blue, while not implemented in other counties, and that's shown here in red. The counties that did not ban trans fat in commercial foods served as a control group by which one could compare the effects of the trans fat ban. Counties that banned trans fat in commercial foods showed a decrease in cardiovascular-related deaths. Thus, there is a positive correlation between trans fat consumption and coronary heart disease. Just as a note, trans fats are often formed artificially in industrial food production. Many processed foods, as shown here, contain trans fats. Trans fats are often formed in industrial food production when manufacturers turn liquid oils into solid fats. More and more companies are responding to the concern that trans fats are correlated with cardiovascular disease by eliminating trans fats from their products. This graph displays a change in the risk of coronary heart disease on the y-axis versus an increase in the consumption for each of three categories of fat on the x-axis. When eating trans fats, the percent increase in coronary heart disease is large, even with small increases in consumption relative to saturated fats. The data from this study suggests that the consumption of monounsaturated fats, such as those found in olive oils, results in a decrease in coronary heart disease. Coronary heart disease results from plaque buildup in the coronary arteries that serve the heart muscle. The plaque is composed of cholesterol and triglycerides and calcium. Fatty deposits along these arteries contain a high proportion of trans fats. This lends evidence to the causal link between trans fats and coronary heart disease. This graph displays a positive correlation between the amount of saturated fat in the diet of people in various countries and coronary heart disease as measured by male coronary heart disease deaths per 10,000 people. Now, correlations are statistical links between two variables, but they should be viewed cautiously. Correlation is not always causation. The correlation between high saturated fat consumption and heart disease does not prove that high saturated fat consumption causes heart disease. For example, we have two countries here with the same percentage of saturated fat in the diet, but very different levels of coronary heart disease. Trials with large sample sizes provide more reliable data about causation. In studies of heart disease among people, there are many variables that might play a role. In trials, it's often difficult to eliminate these variables in their importance in causing heart disease. In fact, other studies have reported an inverse relationship between saturated fat consumption and coronary heart disease. More and more genetic links to coronary heart disease are being unraveled. In other words, in terms of coronary heart disease, genetics may be more important than diet. The next two IB syllabus statements are: identify triglycerides, phospholipids, and steroids from molecular diagrams. Compare the use of carbohydrates and lipids in energy storage. You should be able to identify triglycerides at this point. I need to introduce you to phospholipids and steroids, and we need to compare carbohydrates and lipids as energy storage molecules. Just to refresh your memory, here is a triglyceride. Triglycerides have various roles: energy storage, insulation in mammals, and as a waxy cuticle on the exoskeleton of insects and the leaves of plants. Carbohydrates and lipids both function in long-term energy storage. Lipids have two advantages. Lipids contain twice as much energy per gram as compared to carbohydrates. And lipids are non-polar, hydrophobic, so they are stored with no extra water. Lipid storage is more space efficient in terms of the vacuoles within a cell. Lipid storage is more efficient in terms of the energy stored per gram of body mass. Here's the next IB syllabus statement. State three functions of lipids include energy storage and thermal insulation. Here's another diagram of a triglyceride. Triglycerides have various roles: energy storage, thermal insulation in mammals, and as a waxy cuticle on the exoskeleton of insects and the leaves of plants. However, in terms of the functions of lipids more broadly, we could include phospholipids, and the role of phospholipids is to provide the fundamental structure of cell membranes. I just showed you this IB syllabus statement earlier, but it's back here to emphasize that we need to look at phospholipids and steroids. A phospholipid is composed of a glycerol, seen here, two fatty acid chains instead of three, as would be found in a triglyceride. Instead of the third fatty acid, a phosphate group is attached to the glycerol here. Now, what's interesting about a phospholipid is that while the fatty acid tails are non-polar, hydrophobic, the phosphate group is polar. It's hydrophilic. Phospholipids are amphipathic. As a long molecule, they have a polar head that attracts water and non-polar tails that repel water. Here is another diagram of a phospholipid. I've rotated the molecule to orient the polar phosphate head up and the non-polar hydrophobic fatty acid tails down. Here is a different look at a phospholipid. The glycerol is drawn as a rectangle. The two fatty acid chains are clearly shown as repeating units of CH2, and the hydrophilic phosphate group is drawn as a circle. But take note, this end of the molecule is polar, and this end of the molecule is nonpolar. The molecule is amphipathic. Here is a very simplified view of a phospholipid. The hydrophilic polar head is here, and the hydrophobic nonpolar tails here. So now that you can identify phospholipid structure, here are the next two IB syllabus statements. Describe the amphipathic structure of phospholipids and explain their arrangement in membranes. Explain how the hydrophobic and hydrophilic properties of phospholipids help to maintain the structure of membranes. So here is the fundamental structure of cell membranes. This is a phospholipid bilayer. A layer of phospholipids here and a second layer here, but look at the relationship of the phospholipids to water. The hydrophilic polar heads in contact with water, both outside and inside of the cell. The hydrophobic tails project to the interior of the membrane because they repel water. Thus, nonpolar is more stable with nonpolar. This is nonpolar interactions. In this diagram of a cell membrane, we can see all the components of a membrane, including proteins and carbohydrate chains. But let's leave the complexity of the cell membrane to the cell unit. Instead, let's focus on the fundamental structure of the cell membrane, a phospholipid bilayer. Take a close look. You can see the phospholipids arranged in a bilayer with both water within the cell and outside the cell. The phospholipid bilayer is very stable with good integrity because the polar heads on each side of the membrane attract water and the non-polar tails are oriented away from water. The tails are oriented toward each other in the middle of the membrane. Unsaturated fatty acids have bends in the hydrocarbon chain, as can be seen in this left diagram. Membranes composed of many unsaturated fatty acids will be more fluid as the distance between molecules increases. Plants will manufacture more unsaturated fats in the winter when molecular motion would generally slow due to cold temperatures. As winter temperatures decrease and molecular motion generally slows, the membrane would remain appropriately fluid. The more viscous membranes composed of saturated fatty acids, such as can be seen here on the right, would be more appropriate for warm summer temperatures. And while you've seen this IB syllabus statement before, we still need to take a look at steroids. Identify triglycerides, phospholipids, and steroids from molecular diagrams. Steroid molecules, such as this hormone testosterone, are nonpolar. And while they're not at all like triglycerides, they're often lumped with triglycerides because steroids are nonpolar. Steroids are derived from cholesterol and have this characteristic four-ring structure. When you see this structure, you're looking at a steroid. Cholesterol is found naturally in cell membranes, particularly animal cell membranes, where it reduces the fluidity and permeability to certain molecules. And that brings us to the end of IB Biomolecular Biology Part 5, on to nucleic acids in Part 6.